warahmatullahi wabarakatuh, and hello to everyone. Our topic is about Recombinant DNA Technology. DNA Recombinant Technology, also known as genetic engineering. DNA Recombinant Technology means various procedures and techniques used in gene manipulation.
Firstly is target DNA, sequence of DNA containing interest of genes. For example, gene encode for protein which can dissolve blood clots in heart attack therapy. Next, restriction enzyme. Enzyme that cuts DNA molecules at the restriction site and produce short restriction fragments. Restriction site is the short base pair sequence of 4 to 8 base pair recognized by a particular restriction enzyme. Restriction site contain palindromic, where the same sequence of bases can be found on both DNA strands but in opposite direction. After that, DNA cloning vector.
DNA cloning vectors. DNA cloning vectors are DNA molecules that are able to carry foreign DNA into a host cell. The foreign DNA is inserted into a cloning vector which then replicates into a host cell. For example, plasmid and cosmid. So, we go next to host cell.
Thank you. Next is host cell. Host cell is a cell that accepts foreign DNA in cloning vector and allows it to multiply in recombinant DNA. Host cell can be two types, which is prokaryotic cell and eukaryotic cell. Prokaryotic cell, example as bacteria and E. coli. For eukaryotic, yeast or animal cell. Now, we're going to show you next tool, which is modifying enzyme.
So next is the last tool for the recombinant DNA technologies, which is the modifying enzymes. So the first examples of modifying enzymes is DNA ligase. The functions of DNA ligase, it is used to join up the DNA fragments, the foreign DNA fragments containing the genes of interest and bacterial plasmid by catalyzing the formations of phosphodiester bond. So the second examples of modifying enzymes, it is a Taq polymerase. The Taq polymerase is the short form for the Thermus aquaticus polymerase. It is used to replace the DNA polymerase in polymerase chain reactions, or known as PCR. It is because Thermus aquaticus, it is hot spring and it is the suitable temperature for the PCR to occur. So the Taq polymerase, it adds the nucleotide to the third end of primer to form the new strand of DNA. So that's all for the tools in DNA recombinant technologies.
Now, we will proceed to gene cloning. Gene cloning. When you heard a word cloning, you must think actually cloning is like copying the old organism such as Dolly the sheep. Well, however, clone is something that makes genetically exact copy of it. There are 5 stages of gene cloning. Stage 1, isolation of genes.
Stage 2, cut or clip.
Stage 3, insertion.
Stage 4, transformation and amplification.
Stage 5, screening.
Stage 1, isolation of genes. Isolation of genes have 2 types of cell. The first one is human pancreas cell, and the second one is bacteria E. coli. Human pancreas cell, or known as gene of interest, will isolate from the DNA of the donor cell, and the bacteria E. coli will isolate its plasmid. Now we'll go to the stage 2, which is cut and cleave. So now here we go, the stage 2 of method of gene cloning, which is the cut and cleave. The DNA donor, which is, we know it is from the human pancreas cells, and the bacterial plasmid is cleaved using the compatible restriction enzymes. So when it is cleaved, it makes the single cut within the lag Z gene, which is the lag Z gene, it is the sites of cleaving, and it has two types of cut, which is the staggered cut and the straight cut. As we know, for the staggered cut, we use the EcoRI, which is EcoR1 staggered cut. And for the straight cut, we use the small one. As we know, the long form is Serratia mercescens. So we will cut the plasmid in staggered cut way. That's this, and this we cut it and cut it. Okay, just staggered way to form the sticky end. Sticky end. You can see this. This is the sticky end, the staggered way. Okay, nice. It's nice. So like this, and so this is the staggered cut, and this is the sticky ends. That's all from the cut and cleave. Next is stage 3, which is insertion.
After that is stage 3, insertion. Insertion is DNA fragment or gene of interest is inserted into the plasmid. Plasmids are open right now, and it has to combine with DNA ligase to combine with complementary bases. So we will glue the complementary bases to forming recombinant DNA. DNA ligase will catalyze the formation. So just imagine the glue will act as DNA ligase. At the end of the open ring, plasmid is sticky, so they'll join together and form phosphodiester bond. So I will glue and it already forming recombinant DNA. So the next stages is transformation and amplification.
Stage 4, transformation and amplification. This will result in 3 types of bacteria. First, bacteria that don't take up any plasmid. Second, bacteria that take up non-recombinant plasmid. And the last one is bacteria that take up recombinant plasmid. Following the introduction of recombinant plasmid, the bacteria E. coli must be cultured in a medium that containing amphiphylline and the sugar agar. The bacteria E. coli is cultured in a medium that contains amphiphylline and sugar agar. The transformed bacteria E. coli, which have taken up plasmid, are able to grow and form colonies because they are resistant to amphiphylline. Stage 5, screening.
Thank you. Stage 5, screening. As you all can see, this is petri dish. In this petri dish, it has it contains recombinant DNA, and it also contains agar and also ampicillin.
This is white colony. White colony can survive because there is ampicillin. It cannot hydrolyze agar and cannot change glucose to beta-galactosidase because laser is disrupted. So it maintains the white color. And so this is the blue colony. The blue colony, it grows because it has ampicillin. The lag site is intact,
So it will change the glucose to beta-galactosidase and the glucose will change into color of blue. Performance for the gene method of cloning.
Recombinant DNA Technology. Recombinant panjang sangat ah. Balik.
5, 4, 5, 4, 3, 2.
Aduh, rosak dah mata kita, Mai. Dia jumpa dorang saja.
Kan?
Kau buat apa?
Tak buat apa.
Kau hancur.
To join up the foreign DNA fragments. Ali, kamu juga macam
Gene cloning is a process in which
It also has epicidin and apa satu lagi?
Ayuk.
Epicidin apa?
Ayuk.